Has anyone encountered infection by Vibrio cholera? No? That's good, because now in the world, the hygiene condition is getting better and better. And Vibrio infection is not common. And previously in China, like decades ago, Vibrio cholera infection is the #1 problem in China. And it'll kill a lot of people if infected. Let me show you a little bit information. We cannot finish this today. Vibrio cholera is the bacteria, the spiral bacteria like this. They look like, okay, let me show you. They are gram-negative. So after gram staining, they show this red color or pink color. Okay, I beg your patience. They are, okay, let's check their shape. They are like this coma shaped or curved rod. This one is a curved rod. And some like coma shaped like this, gram-negative. And this bacteria is highly motile because they have one single flagellum on one pole. Okay, this is the bacteria. Okay, on one end of the cell there is one single flagellum, so that which makes the bacteria very actively motile. Just remember the video I showed you, the hanging drop method, to check the motility of live bacteria with microscope. So that's they use the Vibrio cholera. And we can culture the bacteria on TCBS agar. This is agar containing thiosulfate, citrate, bile, and sucrose. That's specifically designed for Vibrio cholera. And on the plate, they will form the convex, smooth, and round colonies. So that's the texture or morphology of the colonies. The bacteria is able to ferment sucrose and mannose. That's carbohydrate fermentation. They produce oxidase positive. And here are important features we want to remember. Vibrio cholera, they prefer a high pH, very high pH, 8.5 to 9.5. And they are rapidly killed by acid. So which means that if you suddenly take in some Vibrio cholera, it's likely that the bacteria will be killed in the stomach. And also it means that if someone is suffering from Vibrio cholera infection, that person must have taken up a lot of bacteria, large number of bacteria, and infect the person. And the bacteria is also halotolerant. Halotolerant, that means that they are resistant to the presence of salts like that. So which means that this bacteria can survive in fresh water, but can also survive in seawater. So sometimes when they check the seafood, they would find some Vibrio contamination in the seafood, like in shells, in fish. Okay, so this is the culture and characteristic. The antigens, the first antigen is H antigen. Why H antigen? Where's the location of H antigen? Flagella, right? Because it has a single flagellum, it's composed of flagellum protein. So it contains the H antigen on the flagellum. And this is a protein antigen, so it's heat labile. It cannot resist to high temperature. Another antigen is O antigen. O antigen, that's the lipopolysaccharide on the outer membrane of gram-negative bacteria. And right now there are over 150 different types of O antigen discovered. But within all these different type of O antigen, only type 1 and type 139 serogroup, O1, O139, are able to cause the classic cholera, the, the fatal disease. The other serotypes will cause mild diarrhea, mild disease. Only these two can cause the cholera. And there is another biotype which discovered recently and which causes the last pandemic all over the world. That's a biotype L2, okay, L2. Okay, the classic contains O1 and O139, while L2 is a new type. It produces hemolysin, this specific, and also the L2 type is resistant to polymyxin B. Any idea about polymyxin B? When we talk about the antibiotics, polymyxin B is a special type which targets on the cell membrane. So we can use polymyxin B to treat gram-negative bacterial infection. But the L2 type is resistant to polymyxin B. The transmission is through the oral route, like contaminated food or water. And we know that only a large number of bacteria can cause infection in a person with normal gastric acid level. And yeah, as a healthy person like you should have a higher level of gastric acid, especially if you have a lot of protein in your diet. You should have a higher level of gastric acid. The bacteria does not reach to the bloodstream. They stay in the intestinal lumen. They are not invasive. They don't invade into our tissue, but they produce a toxin, the cholera toxin. It's very toxic, which causes a problem. The cholera toxin is the exotoxin. Okay, it's an exotoxin contains both the A subunit and B subunit. Actually, it contains okay 5 B subunit and 2 A subunits. And when it binds to the receptor, the GM1 receptor on the epithelial cell in the intestinal tract, it's internalized. Then in the cell, the A subunit is released. Okay. Then it activates an enzyme called cyclase to convert ATP molecule to CAMP. And we know CAMP is one of the most important second messenger. It can further activate protein kinase A. And protein kinase A can open the channel on the cell membrane, the CFTR channel, and that can secrete large amount of water and electrolytes into the lumen, so that a lot of water and salts will be lost. And meanwhile, the cell would transfer more water from the bloodstream into the lumen, so that the person would suffer from dehydration. And that is a major reason why this infection is so lethal. The person would dehydrate in a very short time. So incubation period can be from half a day to 4 days, varies, but the onset of the disease can be sudden. It's sudden onset. And the progress of the disease would be very quick. Some people say that you can see a person walking around on the street normally, but suddenly the person would have a diarrhea, and within several hours, the person might die. So it's very sudden onset. And they will release a rice water stool. That's a characteristic stool containing the mucus, epithelial cells, and large number of vibrios. And they will suffer from dehydration, circulatory collapse, and no urine. This is a picture of a patient in 19th century. Normal. This is the healthy person before the disease. This is the person with cholera, and you can see the typical blue skin color. So we are talking about Vibrio cholera, and so that's, I guess, about here we left last time. And last time we learned the Vibrio biological properties and also their clinical findings, the disease or it causes, and also how to prevent. No, we didn't mention that. Okay. We left here about their laboratory test, how to diagnose cholera infection. The specimens we want to collect are these stools, so that's obvious, okay? And from the stool, you can make a quick check to see the live bacteria. So that's the method we introduced before, the hand drop method. So we can use either the dark field microscope or the phase contrast microscope so that you can see the highly motile bacteria. And that's the typical Vibrio bacteria. And to culture the bacteria, we should keep in mind that the Vibrio prefer a high pH. So no matter what type of agar you use either the peptone agar, the blood agar, or special design TCBS agar, we should adjust the pH to around 9.0 to culture Vibrio. The specific test here, specific means that you can tell exactly it's Vibrio. So that relies on the specific activity of antibody to bind a antigen. So we can use either the slide agglutination test to detect the anti-O1 or anti-O139. So that's the presence of antibody in the serum. So this is the laboratory test. Okay. The epidemiology of Vibrio, I think, yeah, we mentioned this a little bit last time. Vibrio cholera was actually not a global problem. Over 200 years ago, it was confined only in part of South Asia, especially the Ganga River area. So it's only a local disease. But since 19th century, due to the global transportation and global travel and global business, the bacteria was carried all over the world, including Europe, like England, and also East Asia, China, and even America. Yeah, it's all over the world. So since 1817, in just 100 years, there were 6 pandemics of Vibrio outbreak. 6 pandemics. And all these the Vibrio biotypes in these 6 pandemics were the type O1, the classic biotype O1, the serotype O1. There was a last pandemic happened since 1961. That's the 7th pandemic. And this time it's Vibrio cholera biotype Altor. We mentioned this before. It's a special biotype. Okay, so that's the history of Vibrio outbreak in recent 200 years. After infection, the patient, the victim, they would release a large amount of bacteria to the environment. So that's why once there is Vibrio infection, it's not any person's problem, it's a problem of some area. And even when the patient recovered, let's see, the convalescence, they become healthy again, but still they can release large amount of bacteria into the environment for weeks or even for months. And that will contaminate the environment. And we know that the transmission of the, the disease is usually through contaminated foods or water, particularly the drinking water. Right now in the modern world, like in China, like in other developed countries, people are drinking clean water. So the tap water is clean, is already treated. So you use chlorine to kill bacteria in it. Or that at home, some people they prefer to filter the water or boil the water. So that will solve the problem of infection of Vibrio cholera. But in a lot of developing countries, the condition is not well improved. And still they have the challenge to prevent Vibrio cholera infection. So we should try to improve the sanitation, particularly food and water, especially in those developing countries. And the patient should be isolated. And we know that in their waste, there are a lot of bacteria, and we should treat the waste before we release them to the environment. Okay, disinfect the waste before they are released to the environment. For the Vibrio cholera treatment, I think we mentioned a little bit last time that antibiotic treatment is not the first emergent way to help the patient. We want to help the patient by giving them enough water and electrolytes. So that can help them to recover because they are suffering from dehydration and loss of electrolytes. That's the most important therapy. Okay, but yeah, you can also use antibiotics once it's not in the urgent condition. Okay, before the end of this lecture, I'd like to introduce a little bit story of the Vibrio, the cholera outbreak in London. What happened in 1854? It's a story of Dr. John Snow. Has anyone heard of him before? Dr. John Snow or Dr. Snow? No? Okay. Yeah, he was a doctor in London. So that's the story happened in 1854. In the history, before the discovery of microorganisms, for a long time, people in a lot of countries have some theory that some disease is due to the dirty air, the filthy air, which can cause the disease. Especially when they travel from one place to another place, they thought the air in that place is not clean, so that a lot of people would get sick. And there was also a theory of cholera. People believe that for a long time, cholera is due to the dirty air, and they are not related to the water. But this is the story happened in 1854. There was a cholera outbreak in London, in the area close to a street called Broad Street. It's not Broadway, Broad Street. And what Dr. John Snow did is that he tried to visit that area and identify the cases of cholera in a lot of families. And then he labeled all the cases in a map. Okay? all the cases of cholera infection in the map. What he found that, well, there is a cluster, okay? So this infected person are living close to each other in this community. And in this area, in the middle of this area, there was a well, a pump, where people get fresh water from the pump. And well, that gave us some information that maybe cholera is due to the contaminated water. And when people use that water, they got infected. But Dr. John Snow also noticed some exceptions in this investigation. In this area, there was a brewery. You know what is brewery? Brewery. It's a factory which make beer. Okay, so in this factory they make beer, and people living in that brewery, they didn't get infected. And Dr. Snow asked them why. They said, well, we have beer to drink. We don't drink water. So that's why they didn't get infected, because they have a lot of beer to drink. And there was another exception. Okay, there was a family far away from this community. They live like 7 miles away. And that family also got infected. And if it's due to contaminated water, why they got infected? And it turns out that, well, that family used to live in this area, and they adapted to the flavor of this water. So they like the water. So every day they travel a long distance to this pump to get some water home. That's why they got infected. So they use the same water from the same pump. So this is the first time scientists utilize statistics on epidemiology. So make this epidemiology an independent subject. And now we are using statistics all the time to study different type of disease. Okay, either it's related to infectious disease or nutrient limitation, a lot of problems. Okay, so that's the cholera story in London.